This conference will now be recorded.
Synonym: biological source, source, cultivation, collection, and preparation. Microscopic characters, chemical constituents, uses, adulterants, and nutrients, storage. Synonym: fennel fruit, Fructus peniculum, biological source. Fennel consists of dried grape fruit of plant known as Peniculum vigar Miller family. Umbelliferae obtained by cultivation. It should contain not less than 0.6% of anethole calculated on dried basis. Geographical source is indigenous of Mediterranean countries and largely cultivated in Romania, Russia, Germany, France, India, and Japan. In Asia, Java, and Taiwan, it is cultivated in Gujarat, Punjab, Maharashtra, Rajasthan, Uttar Pradesh, and West Bengal. Cultivation: fennel is cultivated by dibbling method. Quality fruit of good germination rate are sown just before the springing, pre-branching of herb, and specific arrangement of leaves in air-like segments on the stem required plenty of space in between two plants or in rows. 4 to 5 seeds are put at a time in holes at a distance of 25 cm in between them. Well drained and calcareous soil in sunny situation is found to be favorable for cultivation of fennel. In India, nearly 90% of phenol production comes from Gujarat alone. The crop is kept free of weeds and provided with suitable fertilizers. When the fruit or ripe crop is harvested and dried in the sun, fruit are separated by crushing. Macroscopic letters green to yellowish brown. Odor sweet, aromatic. Taste strongly aromatic. Size 5 to 10 into 2 to 4 millimeter. Shape straight or slightly curved. It is five-sided fruit in the form of cremocarps with pedals and rarely found in the form of mericarps. Fruit or glabrous with straight, prominent yellow-colored 5 primary ridges are beeped. Stylophytes at the top. It is an arthospermous fruit. Transferally cut surface show two commissural villi and 4 dorsal villi. The embryo is small, abundant in upper end with abundant oily endosperm. Commissural surface of the endosperms are not grooved. Microscopic characters. The special feature of the histological character of fennel are the presence of anemocytic stomata on the epidermis and pericarp of pericarp and the mesocarp containing lignified and reticulate parenchyma. Precursory arrangement of cells on
Manjunath, your voice is not audible. Manjunath? Manjunath, you're able to hear me. Others are able to hear Manjunath?
No, ma'am.
Manjunath,
No, ma'am.
one person was allowed fennel. Shilpa?
Yes, ma'am.
Was only one person allotted for fennel?
Yes, ma'am. Only one person is there. Only Manjunath one, ma'am.
To be seen in the class.
Ma'am, your voice is breaking, ma'am.
Hemant is there in the class?
Ma'am, he is having network issues, so Kavishri will give instead of.
I can't see Hemant. I can't see Tausif. Kavishri?
Yes, ma'am. Yes, ma'am.
Yeah, you can start your presentation.
Yeah. Hello, ma'am.
Yes, will you start?
Ma'am. Ma'am.
Yes.
Hi, I'm Kavishri. My topic is coriander under the guidance of Mamta, ma'am.
This conference will now be recorded. This conference will now be recorded.
And I'm going to explain about tannins under the guidance of Mamta, ma'am. So, in this topic, we are going to study about the history, definition, source of tannins, the process of extraction and isolation, chemical properties and chemical tests, as well as classification, uses, and drugs containing tannins. So, history of tannins. The term tannin is from an Anglo-Norman word called tanner, which is from medieval Latin. It is tannere. It refers to use of oak and other bark in tanning of animal hides to leather. Ellagic acid, gallic acid, and pyrogallic acid were first discovered by the chemist Henry Brancarnot in 1831. Julius Lowe first synthesized ellagic acid by heating gallic acid with arsenic acid or silver oxide. Maximilian Nierenstein studied natural phenols and tannins found in different plant species. He himself, along with George Perkin, prepared ellagic acid from Algarobilla and certain another fruit in 1905. Maximilian Nierenstein proved presence of catechin in cocoa beans in 1931. In 1943, Martin and Singe of paper chromatography, that is, the people who found out who developed the method of chromatography, paper chromatography, provided means of surveying the phenolic constituents of plants and for separation and identification, which contains mainly of tannins. So definition, when we arrive at the definition, tannins are polyhydroxyphenolic compounds which have a high molecular weight that are used for the process of converting hide to leather. Tannins are the product of the shikimic acid pathway, which is one of the basic metabolic pathway, and tannins are produced as a secondary metabolite at the end of this pathway. These are mainly used as an astringent and also used mainly commercially for the conversion of hide to leather. Source. The source of tannins. Tannins are the most widely occurring group of natural substance in different families of higher plants. So they occur widely in higher plants like in plants which are trees and other parts of plant. These secondary metabolites are present in solution form in the cell sap and are also in distinct vacuoles present in the cell sap, present in the plant body. The picture here is shown. Shown here is the bark of the Ashoka tree. So coming next to the extraction and isolation. First, in extraction, no specific protocol is followed for extraction process. Generally, maceration technique can be used for extraction. Acetone is added to extraction solvent to increase total yield by inhibiting interaction between proteins and tannins during extraction, or even by breaking hydrogen bond between tannin protein complex. That is, acetone is mainly used as an extraction solvent to prevent the bond break the hydrogen bond between the tannin and protein. Next, after extraction isolation, simple techniques like thin layer chromatography studies are among the key identity tests in most pharmacopoeial monographs for tannins. High performance liquid chromatography, that is HPLC, is robust, simplest, and uh, rapid and quantitative analysis of compounds offers better resolution and lower limit of detection. So, coming next is chemical properties. Chemically, they contain the mixture of complex organic substances, wherein polyphenolic groups are present in with O dihydroxy or O trihydroxy groups on phenyl ring. Normally, they have fairly high molecular weight, and unlike alkaloids, they are devoid of nitrogen. They form colloidal solution with water, and they are crystalline in nature, non-crystalline in nature. So tannins are uh, form colloidal solution with water, and they are non-crystalline in nature and have a high molecular weight. They are soluble in alcohols, glycerin, dilute alcohols, but are practically insoluble in organic solvents except acetone, which is used for extraction process. In solutions, they show acidic reactions due to presence of phenols. Next is chemical test for tannins. Tannins exhibit some specific chemical reactions. First, solution of tannin precipitates gelatin and alkaloids. This is one of the chemical tests for tannins. Then next, another chemical test for tannins is tannins are precipitated by salts of copper, tin, and lead. Uh, Third is they are precipitated by strong potassium dichromate solutions or chronic acid solutions. Fourth test is they show color reaction with iron salts. Ferric chloride gives bluish color or brownish green color. Potassium ferricyanide with ammonia gives deep red color. Tannins are precipitated by 2% solution of phenazone. The tannin solution being prepared with sodium acid phosphate. The main distinguishing test for tannins is the gold beta skin test. This test is most important for tannins because without this test, tannins cannot be classified easily. So, gold beta skin test is a prototype of untanned fresh skin of an animal and is obtained as membrane from the intestine of ox. This membrane is treated with hydrochloric acid, then rinsed with distilled water, and then placed in tannin solution for 5 minutes. It is followed by washing with distilled water and keeping in ferrous sulfate solution. Tannin imparts brown or black color to the skin. Next comes classification. Classification of tannins it is mainly classified into two types: true tannins and pseudotannins. Uh, True tannins are high molecular weight compounds, and pseudotannins are low molecular weight compounds. Example of pseudotannins is gallic acid. For true tannins, it is further classified into three types: hydrolyzable tannins, condensed tannins, complex tannins. Hydrolyzable tannins is an example of pyrogalol. Condensed tannin has an example of catechol. Complex tannin has an example of flavin-3-ol. Now, hydrolyzable tannin is further classified into galley tannins and elagi tannins. Galley tannic consists of gallic acid, and elagi tannic consists of hexahydrodirphenic acid. So, classification of tannins. The tannins are classified into two main groups based on complexity of their chemical nature and according to their behavior on dry distillation. First is hydrolyzable tannins, which is present and which comes under the true tannins with a high molecular weight. So hydrolyzable tannins, these tannins are hydrolyzed by acids or enzymes quickly. Products of hydrolysis are gallic acid and ellagic acid. On dry distillation, gallic acid get converted to pyrogallol. They respond to ferric chloride solution producing blue color. The example of hydrolyzable tannin are gallotannin in nutgall, rhubarb, clove, and chestnut, tannin from oak, myrobalance, and pomegranate bark. So these are the compounds present in hydrolyzable tannins, that is gallic acid, catechol, gallic acid, glucogalin, pyrogallic acid. The catechol is a type of condensed tannins. Ellagic which we'll see in further slides, consists of the type of nut which is present in gallon, and oak and pomegranate is present in ellagic tannin. Then comes these are also called non-hydrolyzable tannins, uh, and also called as phloba tannins or proanthocyanidins. These are resistant to hydrolysis. That is, when they are subjected to hydrolysis, they do not undergo hydrolysis reaction. These are related to flavonoid pigments as they are found via derivatives of flavones like catechin or flavin-3-ol or flavin-3,4-diol. On treatment with enzymes or mineral acids, they polymerize or decompose into red-colored substances called phlobophene. These are insoluble in water and give brownish red color of many plants and drug colors. On dry distillation, they yield catechol. Tannins with ferric chloride produce brownish green color. The green tea, hamamelis leaf, cinchona, and wild cherry bark, uh, pale and black catechu contain these type of tannins. So green tea, Hamamelias, cinchona, and wild cherry bark, pale and black catechu contain these type of contains condensed tannins. So tannins consists of green tea, cherry bark, cinchona bark, and Hamelias bark. Then come another type of tannin, which is not mainly included in the main classification, is pseudotannin. When treated as subgroup, as they do not these are treated as subgroup as they do not obey Goldbeater's test. That is why these are not considered under main classification. These are low molecular weight compounds. Chlorogenic acid in coffee and Nux vomica, ipecacuanha, and catechins in cocoa are example of pseudotannins. The detection test for chlorogenic acid is carried out by extracting the drug with water and treating this extract with ammonia solution, followed by exposure to air which leads to slowly to formation of green color. Chlorogenic acid in the bottom is is an example of pseudotannin. So these are the plants consisting of pseudotannins: coffee, Nux vomica, Ipecacuanha, and cocoa. Uses of tannins. Tannins medicinally show astringent activity by combining with tissue protein and precipitate them. That is, when tannins are uh, come in contact to skin, they combine with tissue protein and precipitate them, which gives an astringent sensation. Example of plants which shows this kind of activities is Bahera, myrobalan, tannic acid, black catechu, pale catechu, and pterocarpus. Almost all tannins show astringent activity. Tannins are also used as antidiarrheal and antioxidant. Example, amra, or also called as mango. Tannin containing drugs like amla, myrobalan are used as diuretic, laxative, purgative, and in treatment of stomach ache. Tannins are also used as cardiotonic. Example, Arjuna is a plant. It is a tree. Arjuna is a tree. Tannins are also used as uterine tonic and oxytocic. Example, Ashoka bark. Tannins are used in treatment of hypoglycemia. Example, pterocarpus. Tannin containing drugs like amla, bahera are used as an ingredient in Triphala churna and Chyawanprash. Commercially, tannins are used in production of leather, ink, and also as coloring agent in wines. Tannins are used topically in treatment of boils, skin eruptions, and burns. So these are some products which show uh, which consist of tannins. And the picture at the bottom left corner is a Triphala churna which consists of Trada, Baheda, and Amla. These are some commercial uses. The first picture is the process of conversion of hide to leather using tannins. Using tannins. There is and there is a picture that at the top corner that shows the production of ink from tannins. That is, when the extract of tannins undergoes reacts with iron salts and gum arabica, it can be used as writing ink. And the third picture is are the uh, plant products which are used in the which are used in color to the wine production to impart drugs containing tannins. These drugs are not included in the syllabus, uh, 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 but these are also equally important. So uh, uh, the name of the first drug is amla, also called as Indian gooseberry. Its biological source is dried as well as fresh fruits of Amblyca officinalis, and it has family of Euphorbiaceae. Activity
This conference will now be recorded.
Flembin, tannins. It is used as diuretic, laxative, ingredient of Triphala and Chyawanprash. Second